Well, it's my great pleasure now to introduce our final next generation leader speaker, Chris Saha. Chris shares a very strong interest in an area that you've heard about here already in using in vitro human stem cells as a way of understanding human cell biology. Chris has, for many years, been using stem cells as a way of studying cell biology, studying fate decisions, and also using these as models for human disease. His training was at UC Berkeley for his Ph.D., which I happen to think is one of the best places on earth to get a Ph.D., a slightly biased opinion. Following which, this is with David Schaefer and Kevin Healy. After this, he went to the Whitehead Institute and worked with Rudy Janisch, looking at the effect of substrates on stem cell growth. And since 2012, has been a professor at the University of Wisconsin-Madison, both in the Department of Biomedical Engineering and also at the Wisconsin Institute for Discovery, especially working on a theme called BioNates, which I needed crib notes to decode for you, which is the bio-nanocomposite scaffolds for tissue engineering theme. So he's really taken a remarkable bioengineering approach to trying to harness stem cells to be able to really study the formation of organs, for example, cell fate decisions. And if this wasn't enough, he also has sort of a second career as a bioethicist. He holds a joint appointment at University of Wisconsin in the Department of Medical History and Bioethics, and in particular has published on the role of the individual in using stem cells for personalized medicine. So with that introduction, Chris, welcome.
All right. Well, thank you for the very generous introduction, and it's really, truly an honor to be included in this group, and I've had a fantastic time the last 2 days meeting many of you, and in certain ways, among the benefits of this program, I think the thing that's resonated and been most exciting for me is to get to see the Allen Institute in unfiltered ways, I think, as one person put it before, and see the types of questions you're asking, how you're organized, and really the scale at which you're asking these questions and also executing them. So it's really inspiring, and I'll take and learn a lot from this experience. So as you heard, my lab is really interested in human pluripotent stem cells. These are really special cells, and they are defined by two characteristics. One is that they can grow forever in culture, in theory at least, and this has a long history. George Gay in 1951 derived the HeLa cell line, and these is immortalized cell lines that have been very useful and detailed in this very popular book that came out a few years ago. But importantly, human pluripotent stem cells have the second characteristics that HeLa cells do not have. And this is the ability to produce all parts of the body, including the brain and the spinal cord, as well as vasculature and other things that may be useful in the brain. And so one of the kind of major limitations of going forward in this field was that there was only one source before 2007 of where you could find these cells, and that was from human embryos. And due to some pioneering work at Wisconsin and Jamie Thompson's lab, from 1998 to 2007, there's been a lot of work trying to harness both of these characteristics. But in 2007, there was really a watershed moment, and this happened during my postdoc at the Whitehead. And this was the ability to not rely on embryos, and it was to use these induced pluripotent stem cells. So this is a technology termed reprogramming, and what it starts off with is taking some skin or blood or cells derived from urine, and you can reprogram them to that pluripotent state and get those two characteristics that are so important. And from there, bioengineers have a new resource to do all sorts of really cool things. So we'd like to tissue engineer interesting human tissues. We'd like to use it for disease modeling and scale it up to potentially some regulatory science applications. And so this is also big science. Shinya Yamanaka, which is one of the pioneers of this field, is theorized that there's 50 cell lines that are needed to cover the Japanese population in terms of antigen diversity, and that these cells would be immune compatible with 90% of the Japanese population. Similar calculations have been done in the US, and it's about 200 to 300 cell lines, and this is actually proceeding in California and partly in Madison, Wisconsin as well. And so this is a framework where you take these cells at the end here and put them back into the patient. And that's obviously a very long translational process that arguably could take decades, although there's some clinical trials in progress. The more short-term goal and a lot of effort in this field has been to do essentially disease modeling in a dish. And to walk you through this, one of these studies took skin cells from a schizophrenic patient, did this two-step procedure, made these pluripotent stem cells, and then made neurons in a dish in a monolayer, and then look at their connectivity. And they found that when you did this from schizophrenic patients, that the connectivity was different from when you did that from a healthy individual or started with a healthy individual. And so they screened a number of small molecules to change the connectivity, and they essentially said that this could be an interesting platform for drug discovery. EPA is interested in using it as a platform to test neurotoxins. And so you can see that this type of resource is pretty exciting in terms of all of these industrial applications. But as I mentioned, a lot of this work is in flat monolayer culture. And one of the kind of striking and inspiring sets of discoveries last year was this idea that you can go beyond a flat layer of cells and you can go to something called a mini brain. And these are pretty beautiful structures that have complexity to them. So the different colors here, the green is neurons, the red are neural stem cells or progenitors, and blue is everything else, also living kind of cells but undefined. And there are certain hallmarks that this lab in Austria, as well as the late Saha lab, generated in Japan, that saw layered structures, only 2 layers, but still layered, and they call this organotypic. And so one of the main challenges here is how can we really move this forward? And there's a lot of interest in the field in kind of overcoming a number of key challenges. One is that if you tried to do this in a kind of scaled up fashion, you would see a lot of variation from organoid to organoid. Not all of them would look like this, and not all of them would have this type of structure. Indeed, many don't even make neurons at all. And then, secondly, you would have a slow process. So it takes up to a year to make these types of organoids. And lastly, there's limited complexity, and it depends on the question that you're asking. If you want to reproduce a visual cortex, 2 layers is not enough, clearly. And also, these structures are not necessarily stable. Some of these, it's been reported that these shrink over time. So, these set out a number of challenges for the field, and I think what we're stepping back and trying to ask is how could we do this in a more systematic fashion? And what it does also signal, I think, to the field is that there is a innate kind of self-organizing program that if you just let cells grow in the appropriate conditions, you can get fairly elaborate structures. But you also need a level of control to perhaps guide them to make better structures or more interesting structures that you're interested in. And so I've put a balance here between that degree of kind of self-organizing programs that are really, this picture here is from Eric Davidson's book at Caltech, who's really mapped out a number of gene regulatory circuits in the sea urchin that make various cell types. And so this is a stand-in for really a large community of developmental biologists. But regardless, engineers see this type of work as some of the processes that are generating these mini brain type structures. And on the other hand, the studies that I described in the previous slide do heavily rely on highly engineered culture systems like the spinner flask, where the shear rate, the media composition, the matrix, all of these highly determine whether you'll be successful in that experiment or not. And the way that I've come to this question is to think about essentially what's on the inside of the cell, the epigenome and the genome, as things that engineers can now perturb at an increasingly fine level of detail, as well as a number of scaffolds that also chemists and engineers have been able to develop with interesting functionality. And my lab is really interested in this interplay between the inside and the outside to make interesting structures that would be useful to neuroscientists, to regulatory scientists, and other folks. And this is really not an easy question for one lab to tackle, and this is one of the reasons why I'm so happy to be at Wisconsin, and we heard about the bio-nano-composite tissue engineering scaffolds theme, which is essentially tissue engineering, but it's three other labs that share pretty much a common space, and they're listed here, and I'll be highlighting some of the scaffolds that they make. And it's not really just this building where Jamie Thompson and others do a lot of really deep and nice stem cell biology. It's the larger Wisconsin campus. And I think what's unique here is that we have a focus to do advanced manufacturing of stem cells. So what you see here is this video that's on my floor that we share with Jamie's lab. And this is being operated to really pipette various compounds to stem cell cultures and using the latest robotics that are out there. And it's not only the campus, we have, I would argue, very unique partners in industry as well. So Cellular Dynamics International is a key company in Madison that's one of the leaders in this field, as well as to go back to the patient and do clinical regenerative medicine strategies. We have Weizmann Biomanufacturing, and then there's an EPA center now that's starting. So this is all hallmarks and capabilities that we're hoping to integrate with through the work that we're doing. Okay, so to get back to the inside-outside focus of my lab, this is what I mean by outside in terms of what people in my team make on a regular basis. And what I want to point out is that there's a large number of scales. This is at the hundreds of microns to tens of nanometers type of scale structure that we have particular levels of control over. And then inside the cell, I have synthetic biology as an inspiration, which is essentially a new term for genetic engineering. However, there is a key focus on parts, modules, and piecing things together in a systematized way, and using that language of circuits that I think is powerful in terms of how we think about perturbing the system. So if we go back, potentially thinking back to the iPhone analogy to the brain, as soon as you put it into kind of an engineering speak, engineers are excited to hack and break and perturb. And I think if we think about the developmental programs that are in the dish, that are in the cell, making those mini brains in the dish, we have that opportunity to go in circuit by circuit and perturb. And so that's what we're hoping to do in a systematic way in some of the questions that we're asking. And I won't be able to focus on this application where we put cells back into the patient, but synthetic biology, I think as a field in general, is quite excited by doing computation in cells. And eventually, this type of vision of being able to trigger and sense disease in certain ways and have logic functions that get encoded in an output that is seamlessly integrated in your body. And this can be essentially additive and multiplexed and combined in Lego-type fashion. At least that's what the vision is. And these have been actually implemented in some mammalian cells. And so as soon as you have these types of capabilities and triggers, we also have various forms of control that in real time could respond to various events. And so this is, I think, a longer term process that my lab is interested in, but for sake of time, I'm going to focus on the shorter term goal of trying to make intricate structures in the dish. And these are a picture from a culture that we have where we have these organoids that we can image in real time and watch cells divide and grow. And this is just a histone 2B M-cherry marker to mark the nuclei so that we can follow each cell pretty easily. And we'd like to structure this not just as a ball of cells, but array them out in a systematic way such that on the side here, you can see we have this micro printing approach that goes out and gives us very precise micron level control over ligands and particular chemical moieties that we want. So this, in this bottom center panel here, we've printed essentially a PEG non-fouling layer, so this polyethylene glycol, and we have neural stem cells growing in an organoid here that has a GFP reporter that we can image in 3D. And so this is a step, and I think what we're hoping to do is move this forward further. So this video is playing, okay. And so now in the middle, you see that this reporter in this 3D aggregate is one of the capabilities that we have now. But what we're really interested in doing is making that structure more tissue mimetic. And one is that we need to have better control of what cell types we have in there. Neuron astrocytes are kind of the obvious cartoon version of what's in the brain, but there's a whole set of other components that we're interested in making that are not shown in this picture. And all of the diversity that we heard about in terms of neurons is also not shown in this picture. And so how do we achieve that type of cell type control is my first story that I'll tell you about. So how do we induce desired cell types from a mature cell type that you start with, such as skin or something else that's quite readily accessible? So that involves epigenetic reprogramming that I'll tell you about. And the second part of my story is to really think about, once we have those cell types, how do we assemble them in interesting architectures? Okay, so let's start with the first story. As I mentioned, reprogramming takes skin fibroblasts that you can take from a biopsy, infects them with 4 transcription factors, OCT4, SOX2, c-MYC, KLF4. These are proteins that bind DNA and then change the epigenome. And this happens through cell division and weeks of culture. And at the end of the process, you see little colonies that are induced pluripotent stem cells. They have those hallmark characteristics of pluripotent cells and are very similar and arguably indistinguishable from embryonic stem cells. And so this has inspired a whole set of people to think about not only this type of transition to go to pluripotency, but you could take skin cells and make induced neurons, and as well as take skin cells and make oligodendrocytes, as well as cardiomyocytes at the bottom here. So there's a whole class of different cell types that you can induce through overexpression of transcription factors combined with particular culture substrates and media. And so we're interested in how this really occurs, and hopefully the understanding that we get in one of these transitions will give us some understanding on how to control all of these transitions if there's some commonalities. That's the hope, that there's something generalizable here in the mechanisms. And so what we focused on is that first skin to pluripotent transition, and this is a snapshot of what the culture looks like if you start from actually a blood cell and go to induce pluripotent stem cells. And what you'll notice is that there's a lot of different types of cells, and it's not clear which of these have actually been reprogrammed. So we have actually a transgenic marker for pluripotency here, and at this stage, all of these little clusters that look almost embryonic-like are negative for that marker. And if you wait 2 weeks in this culture, you'll actually see that this cluster becomes green, and these actually don't. And the green was the marker for pluripotency. And so how do we understand this type of kind of heterogeneous process, as well as what the culture substrate perhaps could be doing? And what we have been, let's go back. What we've been playing with is trying to understand whether if we push and pull on the cell as well as this nucleus, could it open up particular chromatin domains or influence some of the mechanotransductive signaling that could synergize with the reprogramming factors. And this plate here, or this cartoon here is essentially the printing process robot that I mentioned before, but we've made very thin lines that you can see very flat and perturbed nuclei that we can make at different shapes. In addition, we can add a little bit of 3D-ness to this by putting our cells on nanofibers that we can align to different extents. And so we have a whole library of materials that we can then put reprogramming cells on top of. And the hypothesis that we wanted to test is whether we can find any materials that would accelerate that induction process. And the monomers that we use to make all of these scaffolds are in the bottom right here. I won't go through them, but they're different common polymers that are actually biocompatible. And so we have molecular markers, and we've assayed them through flow cytometry. The marker for our starting state, the skin cells, is CD44. That's along the x-axis here, and that's in red here. And then they go through an intermediate state where they lose this fibroblast identity and then acquire pluripotency identity, and that's marked by TRA160, the cell surface marker as well. And it may be hard to see, but all the fibroblasts are starting off in the bottom right gate, and slowly over time, this is day 7, day 12, they lose fibroblast identity and start migrating up to the pluripotent gate, such that by day 22, in this example, about 7% of cells are been induced to pluripotency. And what we notice is actually a lot of our materials work really well. And the kinetics have a stereotype fashion that follow the same thing that you would see on standard plastic. But we did notice some differences, and actually this is just a marker of the final end state, and the quality of the cells that we get at the end is indistinguishable from what we would get in any other reprogramming process. But we did see some differences, as I mentioned, notably early. And so this experiment has two different timelines where we cultured on our materials library early between day 0 and day 17. That's marked in yellow. And then the flip option is that we cultured from day 17 to day 24 late, and that's marked in purple. And if you look at the data here across various different substrates, you'll notice that some of these effects of the different substrates happen early. So it's between day 0 and 17. And notably, they outperform tissue culture plastic on the left here at least 3 to 4-fold. And so we're following this up. And what we noticed as well is that in day 0 to day 17, others have reported a mesenchymal to epithelial transition. And there's molecules, signaling factors, notably TGF-beta, that inhibit that transition. And so we thought that perhaps that mechanism is synergizing what our scaffolds are doing. And so we added TGF-beta in all of our different substrates that actually erased the effects and the synergy or the acceleration of reprogramming that we saw in our experiments. And so that's where we are at the moment. We're trying to really understand how TGF-beta synergizes with various materials properties. This is a very crude modeling approach, but it is a partial least squares regression model that takes into account the various different materials properties that we have in our library, because it's not that porosity and surface area change independently in our library. They're usually linked, but we could find key cues that we think are coming from outside of the cell. TGF-beta is one that came out of the model, which was comforting, and but then identity and surface area seems to be inhibiting the reprogramming process, whereas this polymer TPU seems to be accelerating. And so these polymers could be doing a number of things. They could be absorbing proteins that help engagement and adhesion. They could be modifying and capturing growth factors that help particular types of signaling. So we're trying to deconstruct that at the moment. So in summary, this first part, in trying to induce a particular cell type, we saw that this nanofibrous fibers could help reprogramming that was dependent on TGF-beta. And what we're looking to do is to really narrow down which of these factors actually, and combinations of them, work with that material to help reprogramming, and whether these lamin-associated domains that are thought to be repressive environments that need to be remodeled during induction change on our library. I want to mention that this work is actually has similar conclusions to a parallel study that happened at Duke, where they looked at smooth surfaces versus gratings that they could control at the nanometer scale. And there, they didn't look at the skin to IPS transition, but they went from skin to neurons. And they saw more neurons and characterized them as being potentially more mature by using some of these patterned substrates. So we're trying to understand what are the common mechanisms, if there are any, and it's just where the field is at right now. But I think we're getting better handle of how we could do this more efficiently and quickly, and how to design our culture environment to get these processes going, to get one cell type versus another. Okay, so the second story that I wanted to share with you was how do we use those induced neurons, or just neurons differentiated from pluripotent cells that we heard about before in the in vitro cell types talk, and assemble them in interesting ways. And so just to refresh your memory about these mini brains, one of the things, goals would be to perhaps expand from 2 layers to more, or even add another layer to this organoid. And you would want to trigger a gene circuit or some sort of signaling factor in a spatiotemporal fashion, such that inside this relatively thick organoid, you have a level of control over a particular developmental process. So we looked at the suite of tools that are out there to turn on particular gene circuits. Actually, Novell talked at the end of his talk yesterday about neurogenin 2, which could be one example of something you want to turn on to produce neurons in one part of the organoid versus a different part. And so I think there's complementary tools here. And what we focused on was to expand the capabilities of small molecules. Of course, light is very powerful, but sometimes you need to have a number of different orthogonal controls over the types of factors and circuits you want to turn on. And so we worked on expanding a very common small molecule called doxycycline, and these turn on the tetracycline or TET-on gene circuits that are very common in both human and mouse cell types. And so what we did was we conjugated doxycycline, which is here in purple, and I don't expect you to go through this chemistry, but the starting nanoparticle core here is a hyperbranch polyester that degrades at low pHs. And we also added polyethylene glycol to make it water soluble. And so what this allows us to do is to add functionality to doxycycline, to have it conjugated with various ligands, get internalized and spatially localized in kind of complex 3D structures. And so what we did here is to just show you what these nanoparticles look like. They're on the order of 50 to 100 nanometers. And what they do is they degrade at low pH. So this is the percent of doxycycline that comes out. And this time scale of 0 to 160 hours actually is pretty tunable. So we can go from hours to days to weeks. And months, we haven't gone to months, but weeks is something certainly we can do with this technology. And so what we checked is whether these actually turn on gene circuits well. And the free doxycycline that's just soluble is in the middle panel here. The top is no treatment. And then we've marked our circuit output that's in our stem cell-derived cells in green in this column here, and then a different output in red over here. And we've looked at many of these images, and we see no difference between at least the level of expression that we can get out of our nanoparticle system. The dynamics can be tunable and changed. And we've assayed that much more closely in flow cytometry that's described in that paper. And we've also seen that in certain cell types that we can trigger gene circuits better, and actually more specifically by bypassing some of the extracellular non-specific effects of doxycycline. And I'll leave the work there. What we want to kind of highlight is that these particles give you some added functionality and don't really sacrifice, I guess, the level of induction. In some cases, it's even better. And thinking through the last 2 days, it could be very well be injected into various spots to turn on gene expressions spatially as well as temporally in certain ways. So we're happy to see where this goes, and we're now thinking of applying this to these types of organoid cultures to turn on and off neurons in one spot or another. And I want to end the rest of the talk with kind of brainstorming and telling you in the directions that we're going with this type of work, and think about what these types of circuits could be. So you heard yesterday about the CRISPR-Cas9 system from Neville, and it's actually a really powerful system as well in our hands. And we're not using it to do genome-wide screens right now. We're using it to modify the genome. And so just to refresh your memory, there's an RNA that's loaded into this protein Cas9 that you need to deliver into the cell. And depending on the RNA sequence, it will find and recognize the complementary sequence in DNA and make a double-strand DNA break. From that break, if you have a foreign DNA donor that can serve as a template for homologous recombination, you can make very specific edits to the genome. And so we've been able to do this and put in an mCherry reporter. We've done the confirmation that it's in the right spot in the genome. This is on chromosome 19. And when you look at these cells versus the unmodified cell line and the one that has the right integration, these are indeed a very nice, homogeneous, reproducible, ubiquitously expressed mCherry marked line. And so we don't want to just look at mCherry in nuclei. We want to look at something that's quite interesting. And there's a number of projects in the lab that are looking to mark cells that are astrocytic versus neuronal, which would very complement, or could complement a lot of the work that's being done here that you just heard about, the various trees that happen during differentiation. We're interested in maturity, so we're interested in marking how old various cell types are, because actually most of the cell types that we make in the dish are quite immature. They have fetal characteristics, and they don't gain, at least in terms of gene expression, some of the hallmarks that you would have from adult animals or adult patients. We're interested in marking various disease genes that get turned on and off during differentiation in various parts of the brain. And we're interested in integrating some sensors or reporters of electrophysiology. And you heard from Viviana yesterday, the Archer technology. This bottom panel here is from Alex Cohen's recent Nature Methods paper that's similar in concept in terms of directly evolving a really interesting sensor that could be used to give out certain readouts of activity. So these are all ongoing projects. I think actually in a short period of time, this will be moved forward by the field, if not by us, by many other folks. The other thing that we're playing with, and Naval also talked about, is to take in and out various human disease gene variants. So we're changing SNPs and watching the effect on neural differentiation or maturation or electrophysiology, all of these things that we can see optically in our little organoid cultures. And this gives you access to enhancers, copy number variants. There's all sorts of things that previously have been very hard to engineer into human lines. And lastly, we're interested in that technology that I mentioned before, that Naval also talked about, of overexpressing neurogenin 2 and speeding up differentiation. And this is a picture where they've made layers of cortex in this kind of organotypic 3D model. This is all from rat cells, and the goal in some of this work is to try to make various types of neurons and use various overexpression of transcription factors that we are starting to understand, that I told you about in the first half of the talk, in a layered fashion in these 3D cultures. So to really summarize the second part of the talk, I've shown you a suite of nanoparticles and micropatterned materials that could give you perhaps the types of architectures that you're most interested in some of these organoid cultures. And we're really rapidly generating a number of different genetically modified lines such that we can do and report on really interesting aspects, hopefully, of neurobiology in the short term. And lastly, I think one of the things that was actually highlighted in one of the team talks this morning is this idea of providing some sort of vascularization to these 3D organoids. Usually, if you look in the middle of these things, they're dead because there's diffusion limitations. And so really trying to scale this up and keeping these cells in culture for a long time requires transport, ideally through a vascular system that you could either hardwire into these organoids or have it develop as the organoid expands in time. Okay, so really, hopefully this gives you an idea of where my lab is going and where Wisconsin is trying to go, where we're trying to integrate and turn on various complex circuits inside of the cell to get these cells to mature to various cell types and induce them to become various cell types. And what we're trying to do is then assemble them using some of the automated and biomaterials tools that we have at our disposal as well. This was not a single-handed effort by any means. This is really brave and talented graduate students and postdocs that have joined my lab, and my collaborators are listed here at University of Wisconsin, as well as a number of them outside of Wisconsin that I talk to quite regularly. I acknowledge really generous funding from Society and Science, the Burroughs Wellcome Fund, EPA, and NSF. Be happy to answer any questions.